Hi everyone, welcome to Bioexplaining. This is the fourth part of our topic, sexual reproduction in flowering plants. Let's begin. In the last video, we have studied about double fertilization, right? Wherein what we saw that one of the male gametes fused with the egg cell to give rise to the zygote, and the other male gamete fused with the central cell to give rise to the endosperm, right? So whatever happens after this double fertilization, they are together termed as post-fertilization events. So what does this word post means? Post means after. That is after fertilization. First, let's look into what happens to the endosperm. So the endosperm development precedes the embryo development. What do we mean by precedes? Precedes means occurs before. That is, first the endosperm is formed and then the embryo development occurs. So the PEC, that means what is PEC? This is the primary endosperm cell. The primary endosperm cell, the PEC, which is 3N. 3N means that this is triploid. So this undergoes repeated cell divisions. And as it undergoes repeated cell divisions, it leads to the formation of the endosperm tissue. Now, this endosperm tissue contains reserve food materials. So what is the utility of these reserve food materials? These will provide for the embryo. So now do we understand as to why is the endosperm formation required before the embryo development? Because the endosperm development will contain reserve food materials. So when the embryo forms thereafter, that reserved food in the endosperm can be used up by the embryo. The primary endosperm nucleus that is present, it will now undergo free nuclear divisions which lead to the formation of the free nuclear endosperm. What do we understand by this term free nuclear? Free nuclear means that the nuclear division will occur but no cell wall formation will occur. That means, say this is a common cytoplasm and inside this common cytoplasm, multiple nuclei will be present, right? So, within a common cytoplasm, we can have multiple nuclei to be present. So, this is known as the free nuclei formation, free nuclear endosperm, right? Now, what will happen is much later, after the nuclear divisions have all taken place, the cell wall formation occurs, right? So, this is what you can call to be cellularization. That initially within the common cytoplasm only all the nuclei are present, but then slowly what will happen, the cell walls will start to form. That means now each nuclei will receive their own compartmentalization. So after the cellularization occurs, you can now call it to be the cellular endosperm. For example, if you consider a coconut, say consider a tender coconut, right? So the water that's present inside, that is the free nuclei. It has thousands of nucleus there. While the white kernel which is present, that is the cellular endosperm. Okay. Next, what we can see is there are two types of seeds based on what happens to the endosperm before the embryo formation or the embryo development begins. First category is if the endosperm is completely consumed before embryo develops. This type is known as non-endospermic or ex-albuminous seeds. Non-endospermic means the endosperm has been completely used up. Nothing is left. So non-endospermic. We can find this in case of beans, in case of peas, etc. And the other one is that the endosperm is still present. It still persists in the mature seed. That is, even after the embryo development has occurred, the endosperm is still present. right? So this can be found in case of castor, in case of coconut. And what are these known as? They are your albuminous seeds. The endosperm is present. Okay. Now, what happens is there are different types of endosperm. What are the different types of endosperm? There are predominantly three types. The first type is free nuclear, which I have just given you a very brief idea of. I'll be talking about it in details. The second is cellular endosperm. Cellular endosperm, free nuclear endosperm. And the third type is halobial endosperm. Okay, so there are three major types: free nuclear, cellular, and halobial. So if you look into this diagram, you'll have a brief idea of what these three are. See, the first one we are telling that is a free nuclear. That means what you have, this is the common cytoplasm, which I have just shown you. This is the common cytoplasm. And what are these? These are all multiple nuclei that are present within this common cytoplasm only. Right. So this is the first one, which is the free nuclear endosperm. This one is the second one, which is cellular endosperm. That means one nucleus is surrounded by the cell walls. Means these are all individual cells that are present. And the third one here is the halobial type, which is a kind of intermediate between the two. So let us look into each of them with certain characteristics of their own. First, let's start discussing about the nuclear type. So in the nuclear type, what happens is the first division of the primary endosperm nucleus and few next divisions are not accompanied by any kind of wall formations. That means if this is the cytoplasm, there will be multiple nuclei which are present. There is no walls separating these nuclei, right? Then the nuclei produced are freely lying in the cytoplasm of the embryo sac. They do not have any compartmentalizations. Okay. Much later, this multinucleate cytoplasm, because multiple nuclei are present, what can I call this to be? We can call this to be multinucleate. This multinucleate cytoplasm will now undergo cleavage. And once it undergoes cleavage, what will happen? It will now kind of look like this, right? That is, each of the nuclei now have proper compartmentalization. Understandable? I hope yes. So for example, we have here coconut water from tender coconut, the example which we've just taken, right? So this is about the nuclear type. Next, what do we have? The cellular type. So the name itself tells you cellular type. That means individual cells are formed. So in this type, there is cytokinesis after each nuclear division. That is, as soon as one new nucleus is formed, cytokinesis is followed and you have compartmentalization of that individual cell. Okay. So the endosperm has a cellular form from the very beginning. That is, from the beginning only, this is the situation. That means one nuclei means one cell. And all the subsequent divisions, they are all accompanied by wall formations. So can you understand the difference? In free nuclear, we have seen that the nuclear divisions kept on happening one after the other. Only much later, the cell wall formations occurred. But in case of cellular type, right from the very beginning itself, the cell wall formations were occurring. Okay, so this we can find in case of petunia, in case of datura, etc. So we are done with the two types of endosperm. First is the free nuclear one. Second is the cellular type. Let's go to the third type now. This third type is the halobial type. So as I told you, it is in between the free nuclear and the cellular one. So we can say that it is an intermediate type between the nuclear and the cellular types. So what happens in this case is, your first division that takes place, there you find cytokinesis. But the subsequent ones are free nuclear. Getting me? So the first time the nucleus is dividing, immediately it has its own cell. The wall formation has occurred. But thereafter, when the nuclei are dividing, the divisions are free nuclear. That means multiple nuclei are present in the common cytoplasm. Right? So the chamber which is there towards the micropilar end, that is usually large, and the chamber which is towards the chalazal end, that is kind of small. So if I just have to give you a rough diagram regarding this, what can I tell you? That first only you will have divisions like this, and then there will be these multiple cells. So if this side, I consider this one to be the micropilar end and I consider this to be the chalazal end, what we can say is the micropilar end side chamber is quite large as compared to the chamber which is present on the chalazal side. So I guess we are good to go with the three types of endosperm: free nuclear, cellular, halobial. Now that we are done with the endosperm, let's focus on the embryo development. Okay, so let's begin with the embryo development. So for a very quick recap, let's just remember that when we studied the structure of the embryo sac, what we saw that there were two sides, right? One is the micropilar end and the other is the chalazal end. Okay, at the micropilar end, we had three cells, two synergids, one egg cell. In the middle, we had the central cell with two polar nuclei, and again on top at the chalazal end, we had three antipodal cells. Right. So two male gametes came in and one male gamete fused with the egg cell. So what happened here was that the egg cell fused with one of the male gametes and that gave rise to the zygote. Okay, so what happened was this egg cell was N haploid, the male gamete was again N haploid, that gave rise to the zygote which is diploid. So this zygote is the structure which develops into the embryo. Okay, the zygote develops into the embryo, right? So embryo develops at which side? It develops at the micropilar end of the embryo sac. This is what I've drawn as the embryo sac. Getting me? So only at the micropilar end you have the egg cell where the male gamete comes and fuses, that gives rise to the zygote. So from the zygote the embryo is formed. Now most zygotes divide only after a certain amount of endosperm is formed. Remember when we were talking about endosperm just a few minutes back, what did we say? That the endosperm needs to form before the embryo. Why so? Because the endosperm has the reserve food materials, has the nutrients which will be required by the embryo. Okay, so this formation of the endosperm before the embryo is an adaptation to provide assured nutrition to the developing embryo. That is to make sure that the embryo has its source of food. Good to go? Okay, now seeds will differ. Some will have two cotyledons, some will have one cotyledon, monocot, dicot. Seeds will differ. But the initial stages of embryo development, that is starting from the zygote, how we go to the embryo, those basic stages are more or less similar in both the dicot seeds as well as monocot seeds. Okay, so what are those different stages in case of the embryo formation from the zygote? Let's see one by one. So first you have the zygote. So the first thing that happens is the pro-embryo is being formed. After the pro-embryo, we have the globular embryo. So why is this term globular? Because the look is like, it gives us a globular, or you can say a spherical structure. The embryo kind of has a spherical structure. After this globular embryo stage, what do we have? We have the heart-shaped embryo. In this heart-shaped embryo, what you can note is that there are some suspensor cells that are present. So it is said that this heart stage embryo, you can say that it is a transition period where the cotyledons finally start to form and elongate. So I can write that this heart-shaped embryo is a transition period where the cotyledons finally start to form and elongate. Okay, so this name is given because the shape kind of looks like a heart, right? Then what we have the mature embryo. So according to your NCERT, the mature embryo stage is given after the heart stage, but there also exists a stage in between. So in between heart stage and the mature embryo, we have another stage which is known as the torpedo stage. Okay, so this torpedo stage is nothing but just this heart kind of deepens. So this heart deepens like this. Okay, so this torpedo stage also exists, but according to NCERT, right after the heart-shaped embryo, the mature embryo has been mentioned. So in the mature embryo, what you can find is you can clearly see two cotyledons which have been formed. So these are the stages of embryo development in case of dicot seeds according to the diagram, since I've shown you two cotyledons here. And the stages of embryo development are together termed as embryogeny. Be it monocot, be it dicot, whatever. Stages of embryo development are termed as embryogeny, and overall these are the common steps. From zygote to proembryo, to globular, to heart-shaped embryo, to torpedo, to mature. I hope this is clear. Now, next, see what happens in case of a dicot embryo. So a typical dicot embryo has this structure. So let us analyze each of these structures one by one. What is present? So in a typical dicotyledonous embryo, you have an embryonal axis. Okay, so embryonal axis means just a line, you can say, kind of the center. So the embryonal axis is present, and along with that, two cotyledons are present. So I've marked here as 1 and 2. Those are the two cotyledons which are present. Now, if you notice very carefully into the diagram, you'll see that the portion of this embryonal axis above the cotyledons, that is termed as the epicotyle. Okay, so these are the cotyledons, right? This 1 and 2. So above the cotyledons, these are the, so this region that is above the cotyledon, this is termed as what? This is termed as the epicotyle. Okay, so portion of the embryonal axis above the cotyledons, that is the epicotyle. And where it will terminate? It will terminate at the plumule. That terminates at the plumule, and this plumule is what you call to be the stem tip. Okay. Now, this cylindrical portion below the level of cotyledon, wherever the cotyledons are ending, below the level of the cotyledons, you see what we have present is the hypocotyle. That is the hypocotyle. Okay. So, cylindrical portion below the level of the cotyledons is the hypocotyle. And this hypocotyle will terminate where? It will terminate in the radicle. what you call to be the root tip. This part, this region here, this part is the radicle or the root tip. This is where the hypocotyle terminates. Okay, and what you can see is right at the ending, the root cap is present, which covers the root tip. Okay, so just going through it very quickly once again, the different parts that you have, one is the embryonal axis which is present, right? And then what do we have? We have two cotyledons that are present. So above the cotyledon level, we have the epicotyle to be present. Epicotyle is present above the cotyledon level. Below the cotyledon level, hypocotyle is present. Where will the epicotyle end into? It will end into the plumule, which is known as the stem tip. And the hypocotyle below, it will end into the radicle, which is the root tip. And the radicle or the root tip, it ends in a root cap. That is, the tip is covered by a root cap. So a typical dicot embryo structure is clear to you all. Next, let's talk about the structure of a monocot. For example, let's consider grass. So this is the diagram overall. So what you know that monocot, this term mono, what does mono means? Mono means one and cot means cotyledon, right? That means that in case of monocot, we just have one cotyledon to be present. And this cotyledon is known as the scutellum, right? So the cotyledon in case of monocot, in case of grass, is known as the scutellum. Where is it situated? It is situated towards one side, lateral side of the embryonal axis. Okay, so let me just keep a note of this, that the cotyledon is situated at one side of the embryonal axis. Fine. Next, what do we have? Straight come below at the lower end. At the lower end, the embryonal axis has what? It has the radicle, the root cap, the coleorhiza. Can you come to this region? Like, can you identify which part am I talking about? This part. Okay, so what are these? At its lower end, the embryonal axis has the radicle and the root cap, which is enclosed in an undifferentiated sheath called the coleorhiza. Right? So right at the end, you have the coleorhiza, which is like the covering tip of your radicle and the root cap. So this is what you have towards the end, right below. Talking about the portion of the the embryonal axis above. So see, there is this level of attachment of the scutellum. Can you understand which level am I talking about? This one, where the scutellum has been attached. This region where I'm pointing to, site of attachment of the scutellum. So above that, what is present? Above that, you see, you have something known as the coleoptile. Right? You have something known as the coleoptile. So what is the coleoptile? Just like below you saw the coleorhiza was covering the radicle and the root cap. Similarly, coleoptile covers the epicotyle. That means the shoot apex and the leaf primordia, which is known as the epiblast. Is it proper? So just for a quick recap, let's see that embryos of monocotyledonous seeds possess only one cotyledon, right? And that cotyledon, what do we call it to be? We call it to be the scutellum, right? So where is it present? It is present on one side, one lateral side of the embryonal axis. At the lower end, we have the coleorhiza covering the radicle and the root tip. And above that, what do we have? Above the site of attachment of the scutellum, you have the coleoptile. What does this coleoptile do? The portion of the embryonal axis above the site of attachment of the scutellum, that is the epicotyle, and this epicotyle is covered or protected by the coleoptile. I guess we are good to go. Okay, that is it on this topic. Please stay tuned with us. In the next video, we are going to talk about fruits and seeds. If you like this video, then please like, comment, share, and yes, don't forget to subscribe to my channel by explaining. Thank you. Bye-bye.